So, okay. We are very interested for many years, more than 20, on two G protein couple receptors that we clone in the lab that were named not very nicely, but just with the number, GPR37 and GPR37-like protein 1. And I don't have to say anything about the G protein receptor family, just that is one of the biggest families of the human genome, and about 1/2 of the drugs in the market are directed against them, because they work as inbox for messages. They have an incredible range of variety. The messenger can be light, energy, lipids, protein, hormones, whatever you have in mind. And in controlling what is outside the cells, they also control an enormous variety of body function. And so you can easily understand why the drugs act on them. But we limit our experience to these two receptors, and we have done a lot of work on the first one. And if you look at the podcast of 2 years ago, I spoke about that. And it's particularly important for people working on neurodegenerative disease, because GPR37 has a role in Parkinson's disease. So you can find either in the literature or in the podcast. And today I will concentrate on the second one. There was also the second one in our mind. So it was always a little bit neglected. But then we continue working on that, and after 10 years, we were not to publish, because at the end was quite interesting. But what is nice is that GPR37 and L1, they are the most closest homologue, but they do something different. And this is like Wolfgang Buss told you. The function of a receptor, although very similar, is very much dependent on which cells it is expressed. So even if the ligand is the same, the structure is the same, the context is very important. And this is a nice example. So like everybody, we want to know the function, and we just follow what the clinic does. So we follow the IMPC rules. This we didn't know when we started, but then it's the same rule that the mouse clinic applied. So it's an approach of reverse genetics, and we decide to start with the knockout mice. We did the Krilox strategy, so I don't have to show you. The gene is very simple, it's only 2 exons, we delete 1 exon. And then we went through all the characterization, thinking that the more we know about a protein, the better. And probably this could also help in finding the functional role. We didn't know which were the ligand till June 23. And we always thought that knowing the ligand, this would make us so happy, because these genes, as you will see, they are expressed in the brain. So we think, oh, probably the ligand is a fancy molecule, NGF, GDNF. And then the ligand was this prosaposin. We never heard about it, but now we learned something also on the ligand. And the ligand now fits quite nicely with what we have found in knockout. So let's start with the protein. It's a normal G protein couple receptor, nothing strange on the structure. And the most closest homologue are the endothelin receptors, so we could guess that the ligand is a peptide or a small protein. The expression is very peculiar, because this is in situ hybridization starting from P4 on. The messenger RNA is only expressed in the cerebellum, and there is a scattered expression through all the brain, very tiny, in the astrocyte. If we go with MAG in situ hybridization, you see a nice staining of this small black blue dot, and the staining is not covering the big cells that are the Purkinje cells Andre was speaking about, but this small cell here are the so-called Bergmann glia cells. This is a type of an astrocyte, specific of the cerebellum. So I just give you an overview of the cerebellum, but you know already from Andres. Just to mention, because I cannot go in many details, that the cerebellum is involved in movement, coordination, balance, and equilibrium, but you are not born with these abilities. In the human, it takes 2 years to develop, as you all know, to develop this coordination and balance. In rodents, and especially rats and mouse, is a little bit faster. So the postnatal cerebellum lasts 25 days. And at the end of that, the cerebellum is perfectly working and has no difference with an adult cerebellum. So if you cut the cerebellum sagittally and you go there, you have already seen with Andres, you have 3 layers in the adult cerebellum that are characterized by different cytoarchitecture that is typical of any layer. So in the molecular layer, you don't have any cells. There are only few cells. We don't mind about them. And there is the enormous dendritic arborization of the Purkinje cells that are the biggest cell of the brain. The cerebellum is nice because it has the biggest cells and the smallest cells of the brain, of the mammalian brain. And these cells, the granule cells that constitute the granule cell layer, are the most abundant cells of all the brain. In fact, if you stay in the brain, you see this nice blue decoration in the cerebellum, and these are the granule cells. It's important to know that the Purkinje cell is the only output of the cerebellum. So without the Purkinje cells, you don't move. I mean, you can start moving, but then you lose your balance and your coordination. So this cell is doing everything. And its activity is controlled quite nicely and in detail. But it's very nice that I spoke after Andres, because I speak about the postnatal cerebellum development like he did. And here I have highlighted the Bergmann glia cells that are these tiny cells in close contact with the Purkinje cell's body. And these are important because are the cells where our receptor is expressed. So schematically, I just recapitulate the postnatal cerebellum development that lasts 25 days in rodents, 2 years in humans. So it's very, very similar. So you start with a different layer organization. In the adult, you have 3 layers. In the developing cerebellum, you have 4. The granule layer is divided in a top part and in a bottom part. And what it happens is that at the beginning, day 1 after birth, the Purkinje cells and the Bergmann glia cells are already there. But there is nothing else. And apparently, the Bergmann glia cells are fine. They will stay like that, but it's not true. Instead, the Purkinje cells, during these 25 days, they grow enormously. They start from a size that is about 5 micron, and at the end, they occupy something like 500 micron. So it's a huge developmental process. What about the granule cells? So they start proliferating in the external granule layer, and the intensity and the duration of the granule cell proliferation is very important because it's the one that determines the function of the cerebellum, because those cells, once they are proliferate, they become post-mitotic. They produce this axon. They attach to the Bergmann glia cells, and they start migrating, and then they reach the internal granule layer where they finish their development. And when they are there, they are mature granule cells. Here they are precursor of granule cells. Luckily, there are many proteins you can use to distinguish between the different type of cells, so you can distinguish proliferating cells from post-mitotic cells. Then we are particularly interested in the Purkinje layer, so we can distinguish with many stainings, and we did with the Bergman glia cells. Here the staining is very nice because it stains both the cell body and the filaments, the Purkinje cells, as Andre also showed to you. And we can also distinguish the mature granule cells from the precursor of the granule cells. And this is very important because we should know what we are looking and which are the differences and so on. This I can just skip. So we were not very lucky because the mice were born and there was no phenotype. And they were great, nothing wrong with them. And if we were in a clinic, this will be put in the database saying perfectly healthy mouse. But we have this one strain, and we want to understand more. So we did many behavioral tests that have to do with motor coordination and balance. And we found the first phenotype that is quite striking, because our mice, they were performing better on the rotarod compared to the wild type. And this is very unusual. Normally, when you take out a gene, they lose some activity. In our case, there was a gain of function. And what was very nice is that our mice, they stay like that all their life. Even when they age, they stay perfectly nice on the rotarod. And we also challenge them changing the speed of the rotarod. So the poor guys, they have to run even faster, but they have no problem, even when they were 12 months old. So we thought, well, if we don't manage and our research is a disaster, we will offer them as an attraction to the Cirque du Soleil, because they are like acrobats. Okay, but we have to go on, you know, we cannot stop. And so just to remind you, this is the expression of the receptor protein. It's there from the beginning. It stays there all the postnatal developmental period, and the staining changes with the time. So it's a little bit different at P5, but then at the end, it occupies all the molecular layer. And it's very similar of the staining you got with other specific Bergman glia cell markers. We look at the cerebellum, and just grossly, we could see anything. They look fine, no problem. But then we went in more detail, and as in the case of Andres, we found that there were alterations in the depth of the various layers, especially in the external granule layer that is highly reduced at P10, and the opposite in the molecular layer. No difference in the internal granule layer. This is just to remind me that we have used a lot of software to analyze the immunofluorescence images. And this is very important because it's not just that we describe what we see, but we can quantify what we see. In the lab, we have many software, but they are all based on software you can find free on the internet, that is ImageJ, and all the ones you buy are just a derivative of those. They can be more fancy, but at the end, this is the base of everything. So we do our observation, but then we can quantify everything, and this is very important. So in this case, we quantify the difference in the thickness of the layer. In the knockout, we have a reduce in the HGL and an increase in the molecular layer. And this could be due to many problems. We found out that the problem was that these cells, they don't die, they just have a different wave of proliferation. And the same experiment as Andres Visto. So we analyze the proliferation by BRD incorporation. And as you can see, there are many differences at different age between wild type and knockout. We quantify the difference. And as you can see, there is the knockout has a different wave compared to the wild type. But the two things, they go together, because while the granule cells proliferate and differentiate, also the Purkinje cell has to do something, as I told you at the beginning. And also in this case, we found the difference between the wild type and the knockout, very clear at all these postnatal days. In this case, we use a nice software that is named IMARIS, in which you can do a 3D rendering of the immunofluorescence analysis, and then you can validate the intensity of the staining. And also in this case, there is a huge difference in the dendritic arborization between wild type and knockout. There is a shift in the temporal reaching of the complete phenotype. And the same is true also for the Berman glia cells, because it seems that the knockout mice, the knockout mice, they reach earlier than the wild type the top of the cerebellum. And this could be considered something like a maturation difference. We don't know. And as I told you, the Purkinje cell is the only output of the cerebellum. Its activity is strictly controlled by wiring with the parallel fiber and the climbing fiber. And we see, just look at the colors. There is a huge difference between the wild type and the knockout in acquiring contact with the fiber that control the Purkinje cell activity. And this is true through all development. At the end of P20, P25, when the postnatal cerebellum is finished, they are similar. And this is also true for the rest of the phenotype. So we decided our knockout mice has to do with proliferation. There are many proliferation pathways. We didn't know which one it was, and so we went through all of them. And at the end, what we found is that the proliferation alteration in our mice has to do with the Sony-Hedgehog pathway. This pathway is very important during embryonic development, but it's also very important in the cerebellum for the postnatal development of the cerebellum. And what is very nice about Sony-Hedgehog is that Sony-Hedgehog is expressed also in adulthood, but nobody knows why. So it's there, and as I show you later, it must do something, but nobody knows what it does in the adult cerebellum. In the postnatal cerebellum development, Sony-Hedgehog is expressed by the Purkinje cells, and it acts on the granule cells, making them proliferating. Without Sony-Hedgehog, you don't have granule cell proliferation. The way this mitogen act is through a quite complex signaling pathway. And what is very strange, it acts through two types of receptors. One that belongs to the same family of GPR37L1, that is named SMUTNET receptor, that is not the receptor for Sony-Hedgehog. The receptor for Sony-Hedgehog is patch, that has more the function of ion transporter. But in this case, patch is the receptor for Sony-Hedgehog. And it's interesting to notice that what is known is that Sony-Hedgehog regulates the granule cell proliferation, but also the Purkinje cells and the Berman glia cells express both receptors, so they are able to transduce the Sony-Hedgehog signal. To cut a long story short, we found a gross alteration in all components of the sonic edge signaling pathways. We look at the protein because we reason that our receptor is on the membrane, so could have difficulties in influencing the messenger RNA level. Probably has to do with protein-protein contacts. And as you see, the major players are very altered, and they reach the same level during the end of the postnatal cerebellum development. We localize patch on our mice cerebellar EP3, so at the beginning of the gross granular proliferation, and we found that our receptor co-localized with patched, so we can understand what is going on, and more interestingly, they do form the co-immunoprecipitate. So apparently they form a functional complex. Just this is published, and the conclusion are that GPR interacts with the patch, regulating granule cell proliferation, but also Bergman glia and Purkinje cell neuron maturation. But now, and this is what we are doing at the moment, we are facing with this problem, which is the functional role of this interaction. And what we don't know is where this interaction takes place, when it is important, when it has a functional role, why is the interaction going on, and especially what is the ligand for our receptor do in this process. So I cannot go in many details, but just to let you know, prosaposin is a neurotrophic factor, but it's a multifunctional protein. It has two major roles. One as an extracellular protein, and in this case, this is the neuroprotectin and the glioprotectin and glioproliferation factors. But it has also a major role in the lysosomal degradation. It helps in degrading sphingolipids. So if you knock out the prosaposin, you have a huge amount of different phenotypes. They are very difficult to understand which is due to which of the function. And as the name say, prosaposin gives rise to 4 different saposins, and each of them has a different role. So it's very difficult to understand something from the knockout. What we look at was the expression of prosaposin in the developing cerebellum. So we look at P5, and we notice, quite interesting, that prosaposin is the red label here, and is produced by Purkinje cells, and the red cells are the Bergman glia cells where our receptor is expressed, so they are there at the same moment. And what is in the literature is that prosaposin is highly expressed during postnatal cerebellum development, but then has no role in the adult, unless there is something happen in the cerebellum, like stroke, something like that. In that case, the prosaposin level, they rise again. And this fits quite well with the expression of the receptor we find. So it seems that the receptor and the ligand are in the same place and at the same moment. So for sure, they have a role in this period. Now I arrived to this wonderful organelle that I met, and I really like them. I like it a lot. Because this slender protrusion of cell is a wonderful organ. And I want to convince you about this. What is nice is that every single cell in your body has a primary cilium. And they protrude very slightly for the cell membrane because they are very short. They can reach maximum 10 micrometer. Normally they are 2 micrometer. And they have a diameter of nothing less than 1 micrometer. They are not motile, So what do? They work as a chemical sensor, signal transduction, and they control cell growth. Here I just received this wonderful software, so I applied to an image, a confocal image I have, and I want to show you that you can, no, I will not show you. Anyway, what I want to show you is that you can label with different proteins either the length of the cilium or the base of the cilium, and this was a 4D rendering, so you can also see how they are bent in the space. To go in more detail, this is the structure of the primary cilium. It all very well remember the motile cilii. The only difference is that they don't have the two microtubule pair at the center, and this is why they are immotile. They have the basal body that is a modified centrioles, and what is very important is that here they have this that is called transition fiber, because there is no protein translation going on in the primary cilium. The cilium has to take from the cell whatever it needs, but it has a sophisticated way of taking things, so it has an active transport that is either anterograde or retrograde for things that have to go out of the cilium. And at this level, there is a selection, what could be in the cilium, what is necessary for the cilium, and what is not. So if you don't pass the examination here, you don't enter the cilium. And this is an electron microscopy images where you can see all the different parts. There are nearly 50 human diseases that goes under the name of ciliopathies that are associated with the primary cilium. And as you can see, they affect almost any organ because, as I told you, the primary cilium is all over. And what is nice is that many of the patients, they have more than one disease. And normally they have polycystic organs. The polycystic kidney disease is one of the ciliopathies. But not only that, normally they also have brain malformation, craniofacial malformation, and recently also obesity has been found due to ciliary dysfunction. How this correlates with the results I have showed to you is the fact that many ciliopathies show defect in the Sony-Hedgehog signaling. And we know why. In the absence of Sony-Hedgehog, the receptor for Sony-Hedgehog is on the primary cilium, and it blocks the activity of the SMUTNED, that is the G protein couple receptor. In this way, the signaling cascade converts on a GLI repressor and the transcription is off, so you don't have proliferation. When Sony-Hedgehog is there, the situation changes completely, so Sony-Hedgehog completely reassembles the two receptors on the cilium. Patchet goes away, while SMUTNED enters the cilium, activates the signaling cascade that converts on GLI, that now is an activator, and the transcription goes on. And in this case, Sony-Hedgehog bound to patch is either delivered to endosome or is delivered to lysosome for degradation of both. What we found in our mice was that if we just look at wild-type mice, so no mutant here, as you expected, the green small things are the cilia. Can you see them? Yeah? So as you expected, the cilia are in the proliferating cells, the granule cells, and they stay there till when the cells proliferate. Then, as I told you, in the adulthood, this layer is not there any longer, and the cilia are not there any longer. But what was very nice is that we noticed that the cilia are in the Purkinje cell layer, and they stay, even in the adulthood. What do they do there? And they are located both in Purkinje cells, and this was already known from the literature. But what we notice is the majority of the cilia are on our cells, on the Bergmann glia cells. Where is our receptor located in the view of the primary cilium? Till now, we could only localize the receptor at the base of the cilium. We were never able to visualize the receptor in the cilium axon M. I have to tell you that the cilium is plenty of G-protein couple receptor. Because it works as a chemosensor, so it has to do with the receptor. So we have decided to change strategy, but before I want to show what we think is the role of our receptor. We think that our receptor has to do with the patched trafficking. So I just have done a sort of an animation, so they interact, and this we have shown. And then we don't know whether the receptor has to do with the localization of patch, either in the endosome, but in this way, both of the receptor will be recycled back, and in this case, there will be a stop of the sonic action signaling, or we don't know whether our receptor is helping the degradation of the pathway of the receptors. And in this case, you just go back to the beginning when you don't have the ligand activator. And the prosaposin could have a role in the trafficking of our receptor, and so in facilitating or inhibiting the contact with the patch receptor. So what we want to do now is that it's very difficult to work with the entire cerebellum, so we want to go with primary culture derived from wild type and knockout, and in this case we can understand the role of prosaposin, where prosaposin has a role in maturation of the Bergmann glia cells or in proliferation, because there are rumors that the Bergmann glia cells could be the stem cell of the cerebellum. And we can also analyze the sonic action signaling, whether it occurs in the presence or in the absence of the primary cilium, and if this has to do, and this is our hypothesis, with the two receptor trafficking. We have also another project, because when there is an alteration in the sonic adjoc pathway, as you can imagine, you have uncontrolled proliferation. And this has to do with the terrible cancer that affects many children at birth, and is the cancer of the cerebellum that has the name of medulloblastoma. And what is important to notice is that having to do with proliferation, each single cell in the cancer has a primary cilium. So for sure, primary cilium has a role here. I need to thank the people who did the job, Glauco that is here, and Chiara, Gina, and Raffaele, who really helped a lot in all the characterization of the knockout, as also Elisabetta and Silvia did, especially for the behavioral part. And then this Sima is an Iranian student that is working on the medulloblastoma project. And I leave you with this. I don't know if this one will go. No. It's a pity. It was another movie. Anyway, I just want to remind you that we like the primary cilium, and especially we like the fact that the primary cilium is on Bergmann glia cells. So.